Dear learners, in this lecture I am going to talk about a very interesting topic, that is plant hormones. As you know, plants are multicellular organisms and their cells are highly specialized or differentiated. For example, some are epidermal cells which serve as protective cells, some are associated with xylem and phloem tissues. Some are guard cells, some are parenchyma cells or collenchyma cells. So, plant cells must coordinate with each other as the specialized cells are not independent packages. For example, a pair of guard cells at the time of their formation, they must coordinate with each other about where the pore or stomata will be formed, because the stomata will only be formed if the guard cells are facing each other, they are face to face, so that the pore or stoma is formed between them. Likewise, differentiation of sieve tube requires coordinated differentiation of companion cells that are associated with sieve tube members. So, plant cells must coordinate with each other. And this coordination is provided by certain chemical messengers, which are called as plant hormones. Hormone is a Greek word that means to excite or to arouse. Originally, they were studied in animals, but soon they were also discovered and extensively studied in plants. Plant hormones are substances other than nutrients that affect growth and development of plants. So, they affect many processes: growth, development, reproduction, longevity, and even death of organs or whole plants. Plants have a genetic blueprint for morphology and development, but they also need to respond to fluctuating environmental conditions. For example, stomatal opening and closing in response to light or the stimulus of water. So, plants can perceive things, they can control things, and they can respond. Even plants are more resilient than animals. They are attacked by herbivores, by insects, by fungi, but they have the ability to regrow their organs as well, and they can survive in a variety of environments. So, plants can perceive things, they can control things, and they can respond. By perception, I mean information about the environment. This must be perceived by plant. Plants must sense changes in temperature, moisture regime, day length, or at cellular level changes in the neighboring cytoplasm that is to be sensed by the nucleus. Then plants need transduction, that is, the information must be transduced or changed to a form that can be acted upon or transported. Then plants must be able to respond. Plants must enter dormancy, produce flowers, or alter type of leaf production depending upon the environment in which they grow or depending upon the stimulus they receive. So, almost all plant communication is by a slow mechanism, that is, the transport of hormones through the plant. Unlike animal hormones, plant hormones are produced in many locations and they have no specialized organs. as we animals have glands for secreting hormones. Similarly, uh, plant hormones are multifunctional, unlike animal hormones, which are very specific. In plants, development is highly sensitive to environmental influences, and plants have no nervous system. So, think of hormones uh, or think of plants analogous to computers. So, think of computer as a plant and its commands as hormones. For example, we have different commands for open the file or to save the file or to print it. These commands are actually hormones, and computer programs uh, as they respond to different commands. For example, if we give the command for print, the computer programs or subroutines will direct the printer to print a page. So that command is hormone, and hormones are thought to activate programs in target cells. So plants can perceive things, control things, and respond, and they communicate, uh, their cells communicate by a slower mechanism, that is the transport of hormones through the plants. So this intercellular communication is brought about by chemical messengers called as plant hormones. In this lecture, I'll discuss five major classes of plant hormones, that is auxin, gibralins, cytokines, abscisic acid, and ethylene. To start with the general characteristics of plant hormones, they are defined as naturally occurring organic substances that at low concentration exert profound influences on physiological processes of plants. Their site of action is usually different from site of synthesis. So they are produced in one part of the plant and then transported to other parts for initiating a response. However, they can also affect the cells in which they are produced. Almost all cells can uh, produce hormones, but certain cells or tissues produced enormous quantities of hormones, and others produce undetectable amounts of hormones. Hormones have low molecular weight and act as a very low concentration, usually in micrograms or micromoles. Hormones are synthesized and stored in regions of transduction and are released for transport through either phloem or mesophyll and cortex cells when the appropriate stimulus occurs. Hormones are mobile both over short and long distances. So their movement over short distances through diffusion or diffusive process, and over long distance they move by mass flow. At the site of response, hormones bind to receptor molecules. These receptor molecules are special cellular proteins which are usually located in the plasma membrane, and there they trigger a response. So hormone does not act at its own. It can act only when it binds to a receptor molecule. So when it reaches a target cell, then it binds to receptor molecule. But if it is not a target cell, it will not respond because that cell lacks receptor. So many organs which are not target do not respond to hormone because they lack receptors. and some hormone functions occur in the same cell in which they are synthesized. I already talked about this. So plant hormones affect not only growth but also processes such as development, reproduction, longevity, and death, which differentiates them from uh, plant growth regulators or plant growth substances. I'll talk about this later. Plant hormones have a simple structure. It was believed that hormone structure contains the necessary information for response. But now there's evidence that hormones only serve to activate the response. Actually, it is the receptor or cell and its nucleus which contain almost all of the information necessary for response. So, hormone just activates or triggers the response. Another interesting thing is that one hormone can perform many functions. So we will discuss later when we discuss different classes of hormones. And many hormones can perform a single function. For example, flowering is thought to believed by uh, to be affected by a combination of hormones. Similarly, hormones are not growth promoters or inhibitors, as often they are classified as some are growth promoters and some are growth inhibitors. It is about the response that can be growth promotion in one region or inhibition in the other region. For example, auxin is thought to be a growth hormone. However, it promotes growth of the apical buds, but at the same time it's also an inhibitor for the lateral buds, that is the phenomena of apical dominus. Another example is of abscisic acid that has been historically thought as a growth inhibitor, but young tissues also have high abscisic acid levels. Now we know that, and exogenous application of abscisic acid can restore normal cell expansion and growth by reducing transpiration rate of the plant so that plants does not run out of water. And hormones can also interact with each other. The For example, the arrival of auxin at target site often causes that site to produce ethylene, which is a gaseous hormone. This ethylene, being a gas, diffuses rapidly and triggers response in the adjacent area more rapidly than auxin itself. So this is an example of how hormones act. So, and how ethylene makes auxin more effective. So these are some of the general characteristics of hormones, that they are endogenous, they are produced within the plant body, they are effective at minute concentrations, their site of synthesis is usually different than their site of action, and one hormone can perform many functions or many hormones can perform a single function. And hormones are not growth promoters or inhibitor. It is about the response that can be growth promotion or growth inhibition depending upon the target cell. Often plant hormones are considered same as plant growth substances or plant growth regulators, but these are two different terms. Plant growth regulators or plant growth substances are synthetic or natural substances that affect the growth of plants. For example, humic acid, fulvic acid, amino acids, benzyl adenine, etc. And many synthetic compounds mimic the effects of hormones, but for clarity, only natural compounds are considered as hormones. They are endogenous, they are produced by the plants themselves. So, plant growth regulators or plant growth substances is a term that is used for hormone like compound that have hormone like activities, and they can be natural or artificial, for example 2,4-D or naphthalene acetic acid. Let's now dive into different hormone classes. So, to start with, uh, I'll talk about auxin, that is the first hormone to be discovered in plants. The word auxin is derived from a Greek word auxin, which means to increase. Uh, this was the first hormone discovered in 1926 while studying positive phototropism in oat seedlings. Indole acetic acid or IAA is the only naturally occurring auxin, although many artificial compounds with indole acetic acid like activity are present. Indole acetic acid precursor is amino acid tryptophan. So indole acetic acid is produced from an amino acid tryptophan. Indole acetic acid is synthesized most actively in the shoot apical meristem, young leaves, and fruits, but it is also present in root tip and is believed to be transported from shoot tip to root tip. The concentration of indole acetic acid in plants is controlled by destruction, that is by removal of side group and oxidation of the 5-member ring. Second phenomena is by conversion to inactive forms by conjugating it to various compounds. Conjugation means attachment. In conjugated form, it is safe from destruction and can be stored in seeds or transported from cotyledons to epicotyle during seed germination. Auxin is transported within plant by two mechanisms, by phloem and by polar transport. This is the only hormone that is transported by polar transport. In shoots and leaves, auxin moves basipetally, that is from the apex to the base of the plant. And in roots, it moves acropatally towards the root apex. This movement is about 11 millimeter per hour, regardless of whether the tissue is in a vertical, horizontal, or upside down orientation. By means of this polar transport system, auxin movement through the plant can be maintained independently of the variation in phloem transport caused by changes in sink and sources for carbohydrates and minerals. So, auxin transport mechanism in plants is via phloem or via polar transport. If we talk about plant responses to auxin, these include suppression of abscission. Abscission is the development of an abscission layer following which the leaves or fruits may fall off the plant. Apical dominance, that is the bud at the tip of the plant or apical meristem, uh, suppresses the growth of lateral buds. So if you want to control the height of plant, especially in horticulture, they often do excessive pruning. So if you cut the plant from the top, then you can make it grow more horizontally or bushy type growth. You can make it more compact, but you need to repeat this process, because if the apical bud is removed or topmost bud is removed, then one or two lateral buds become apical buds and start suppressing the other lateral buds. Other plant responses are cell elongation. It causes elongation of cells, a well-known phenomenon. It is involved in formation of roots in cuttings, fruit maturation, tropic movements, and in xylem different. So these are plant responses to auxin. The next important class of hormones is gibberellins or GAs. More than 135 gibberellins are known, which are numbered as G1, GA1, GA2, GA32, GA135. GA3 is called as gibberellic acid, that was most extensively studied as it can be easily isolated from fungi and used in various experiments. But now it appears that GA1 and GA19 are more active and important in plants. Gibralins have diverse function, but they also have a unifying structure, that is the Gibralin ring system. Gibralin's metabolism is complex and occurs in all parts of the plants, but seeds, roots, and leaves are especially important. Only a few gibberellins are known to be active as hormones. Others are precursors or intermediates in transforming one active form into another. And the relative concentration of the various gibberellins changes in response to environmental signals. For example, when spinach is exposed to long days, the level of GA19 undergoes a five-fold decrease. GA20 and GA29 increase drastically, but GA17 and GA44 do not change. As a result of these changes, spinach stems begin to elongate. Plant responses to gibberellins include conversion of some plants from juvenile to adult conditions and convert other species from adult back to juvenile conditions, involved in flowering, release seeds and buds from dormancy, that is the state of inhibited germination of seeds or buds, stem elongation, and stimulation of pollen tube growth. So these are plant responses to gibberellins class of hormones. Next important class of hormones is cytokinins. Cytokinesis means cell division. So this class of hormones is especially known for promoting cell division. If we add cytokinin to tissue culture medium having auxin and sugar, then cytokinins cause cell division. Carnitine was the first discovered cytokinin, although that is an artificial cytokinin. Natural cytokinin are zeatin and isopentenyl adenine. So chemically, cytokinin relate to purines or adenine. And the most active compounds have a side group containing 4 to 6 carbon atoms attached to the carbon number 6. If the side group is longer or complex, it does not act like a cytokinin. So lacking the proper shape and charge to bind with the cytokinin receptor molecule prevents it from acting as cytokinin. The important plant responses to cytokinins are bud activation, cell division, fruit and embryo development, to mimic the effects of phytochrome and red light in several cases, and to prevent leaf senescence. Next in line is abscisic acid. This class of hormones contains the single compound abscisic acid, which was discovered in species Sycamore. Despite its name, abscisic acid is not a major regulator of abscission. Abscission is now believed to be primarily controlled by ethylene. Abscisic acid does not control abscission directly. The presence of abscisic acid in abscissing organs reflects its role in promoting senescence or stress responses, which uh, precede or happen before abscission. Abscisic acid is widely regarded as a growth inhibitor causing dormancy of buds or seeds. However, dormancy is not just growth inhibition, but it is a complicated set of changes that prepare the plant or seed for advanced conditions or stressful environments. And abscisic acid is especially involved in stress resistance. For example, heating leaves, waterlogged roots, soil dryness due to drought, chilling, and high salinity conditions all have been found to cause sudden increases in abscisic acid level. And if healthy plants are pre-treated with abscisic acid, they become much more resistant to stressful environments. Abscisic acid has a dominating role in stomatal closure under wilting conditions. Its concentration increased from 20 microgram per kg to 500 microgram per kg fresh weight, and plants close the stomatal pores. When abscisic acid is present, other mechanisms causing stomatal opening become ineffective. So, abscisic acid has a dominating role in closing off stomata. Abscisic acid biosynthesis mainly occurs in plastids and partly in the cytosol. The stimulus for abscisic acid production under soil dryness condition is detected by roots. At critical moisture conditions, abscisic acid travels through the xylem to leaves where it causes stomatal closure, but leaves can also produce abscisic acid. Abscisic acid can be removed by being converted to phasic acid, which has no hormonal activity. It is very important to regulate the concentrations of hormones within the plant because overproduction of hormones can sometimes cause damage. Plant responses to abscisic acid include initiation of dormancy, resistance to stress conditions, stimulation of growth at very low doses. So, at low doses, it becomes a growth promoter. Stomatal closure, and probably it is not involved in abscisic acid in abscission in most of the species. So its name is a misnomer actually. So this is a wrong name. The last well-known group of hormones that I'm going to discuss is ethylene. Ethylene is the only gaseous hormone which has the simplest structure, as you can see here. The climacteric fruits which can ripe even after harvesting, for example, mango, banana, tomato, avocado, etc. The changes in climacteric fruits near maturity or near final development are stimulated by ethylene. These changes include conversion of starches to sugar, the cell walls which are broken down and softened, development of flavors and aromas in ripening fruits, and changes in color. Ethylene stimulates these responses, but at first, ethylene concentration is low. As the time progresses, due to positive feedback system, ethylene production increases manifold, that is exponentially. If we talk about transport mechanism, ethylene being gas moves by diffusion. In non-climacteric fruits, uh, which must ripen when they are still attached to the plant, such as oranges, lemon, and cherry. The ethylene concentration remains stable. The plant responses to this gaseous hormone include formation of parenchyma in submerged roots and stems, fruit ripening and abscission, initiation of root hairs, and production of latex. Now that we have discussed different classes of hormones, let's talk about their commercial applications. If we talk about commercial application of auxins, they act as weed killer at high concentrations. They promote growth to such extent that plants are unable to maintain that growth rate. So hence they act as weeds. For example, 2,4-dichlorophenoxyacetic acid or 2,4,5-trichlorophenoxyacetic acid and dicam are well-known weed killers, weedicides. Uh, they also act as rooting hormones, for example, indole butyric acid and naphthalene acetic acid. 4-CPA is a Auxin like product that is used to increase flowering and fruit setting. Naphthalene acetic acid is used for flowering in pineapple due to auxin induced ethylene production. And naphthalene acetic acid can also be used for preventing pre harvest fruit drop in apples and pears. So you see that the same hormone has these opposite effects. These opposite effects are brought about by the timing of auxin application. And this is one of the problems that is very difficult to select and to apply in a right way these plant growth regulators or plant hormones. So we need to be very careful while applying them commercially. Commercial application of cytokinins includes storage of green vegetables to reduce yellowing, and in tissue culture, high oxygen and low cytokinin conditions give rise to root development, whereas low oxygen and high cytokinin conditions encourage shoot development. So they are used in tissue culture. Cytokines are used to induce bud and root formation in callus and to promote lateral buds in tea plantations by suppressing the apical dominus. The commercial application of Gibralins includes seedless grape production, enhancing germination and early seedling emergence in apple, citrus, peach, etc. Production of hybrid seed in cucumber through formation of male flowers, and to speed up malt production. Interestingly, anti-gibberellin compounds are used in ornamental plants for height control, as gibberellins promote extension in height. The anti-gibberellins are used for controlling height. They can also prevent lodging in wheat by producing a short and stiff stem. And they also reduce the need for pruning of vegetation under power lines. So if you have trees under power lines, instead of extensive pruning, you can spray gibberellin. An important commercial application of Gibralin is its use as antitranspirant. Application of minute quantity of abscisic acid to leaves will reduce transpiration to a great extent through partial closure of stomata. So this conserves water and reduces the requirement of irrigation. Although in this process photosynthesis is also reduced, but that is reduced to a lesser extent, about 14%, as compared to transpiration that is reduced by 56%. Abscisic acid can also be used to induce flowering in short day plants, for rooting of stem cuttings, and for prolonging dormancy of buds, seeds for effective storage. Ethylene has many commercial applications. For example, for uniform ripening, which causes abscission in various crops, including cotton, cherry, walnuts. So we can apply ethylene for uniform ripening in these crops, which will cause abscission and they will ripen at the same time. Ethylene can also synchronize flowering and fruiting in pineapple, which makes the harvest of pineapple easier. Ethylene can be drawn out of unripe fruits by storing or transporting them in vacuum, and on reaching market, the air pressure is returned to normal. So, ethylene accumulates again and causes ripening of the fruit. Fruits may also be treated with 2-chloroethyl phosphonic acid, which breaks down and releases ethylene. And in many cases, ethylene acts as Auxin effector. The arrival of Auxin at target site often causes that site to produce ethylene, which diffuses more rapidly than Auxin itself and triggers responses in the adjacent areas that the Auxin aims for. Ethylene also induces parenchyma under submerged conditions. Being gas, ethylene can escape through stomata or lenticles and helps in detecting flooding stress. Ethylene builds up in submerged species and induces the formation of parenchyma in some of the species. This is all about the five classes of hormones. Some of the lesser known hormones are brassinosteroids, jasmonic acids, salicylic acids, trigolactones, and peptides, which are not the topic of this lecture. So, in this lecture, we discussed general characteristics of hormones, how they differ from plant growth regulators, what are responses of plants to application of hormones, how they are synthesized or transported within the plant, and what are the commercial applications of different plant hormones. And ultimately, all of our feelings are due to hormones. This marks the end of this course Agron 508. This was the last topic that we wanted to discuss. Thank you very much for your attention.